And thank you very much for accepting the invitation.
Yeah, and thank you, Antonio, for inviting me. That was nice. It was actually the quickest turnaround ever. Antonio contacted me one day. Do you want to give a talk? Yeah, sure. Can you send me this stuff? Yeah, sure. Do you want to do it in a few days? Okay, good. We could do it in a few days. Okay.
Thank you.
Yes.
Yeah.
Yeah. Show up anyway, always a few minutes later. But
Yeah, I can.
Okay.
yeah, maybe you can first introduce this seminar.
Yeah, you can do.
I can do it well. Yes.
Because I'm gonna manage this.
Right. So together with Sadek and Alireza, right?
Yes.
Yeah, we run this Biomed Journal Club, and the purpose is to share the scientific word, to spread scientific word, because I believe that sometimes articles just get published, and it's not that often. I mean, that we go deep inside in scientific part. Often we just see the article and we don't see it anymore. And I think that it's a shame, because sometimes a very nice study can be overseen. So the topics here, they're quite broad, and mostly it's tissue engineering or artificial artificial biosystems, or sometimes we have invited speakers like you. So and then the topic might be a little bit adjusted, the topic of the seminar. Yes, and how many years it has been so far? Like 1.5 or so?
More than 2 years.
Yeah, so it has been already more than 2 years. As you might notice, we are not yet so visible, but I believe that we have had very nice speakers, which everybody can see on our YouTube channel. And the speakers might choose whether they don't want to be recorded, but we encourage to record and then upload, especially if the studies that you show has been already published. And today we have Martin Hanczyc from University of Trento, who I know personally. So Martin is an associate professor in the Department of Cellular, Computational, and Integrative Biology at the University of Trento, Italy, and a research professor at the University of New Mexico in the Department of Chemical and Biological Engineering. He has published in the area of droplets, complex systems evolution, and the origin of life. And in that matter, in 2011, I know Martin gave a TED talk entitled The Border Between Life and No Life, which everybody can see. Currently, he heads the Laboratory for Artificial Biology, developing novel synthetic chemical systems based on the properties of living systems. And he also coordinates an FET project on artificial cells. So today you would like to talk about one of your papers from Scientific Report that published a few years ago. And yeah, the topic of your talk is artificial cells as biological cell transporters. And now I will just pass you the screen. It's yours.
Great. Can you hear me all right?
Yes. Yes.
Okay, thank you. Thank you, my friend Antonio, for the invitation and for the introduction. And nice to meet you, Sadek. Thank you for inviting me. Very nice of you guys. I'll start by sharing my screen. Oops, it does say I need permissions. Something didn't work out.
Yes. Sorry. Yes. Here we go.
Oh, yeah, that should work now. Okay.
Super.
Can you see my slide?
Yes.
Yes.
Is it in full presentation mode or some other?
Exactly. Yeah.
It's good. Okay, cool. Thank you. Great. So, yeah, I'll speak with you guys kind of more informally about some recent work we're doing. It is going to be, as Antonio said, about droplets, but maybe it'd be interesting for you who are doing some tissue engineering, because we use some of these principles as well, as you will see. And so I will speak on the area of artificial cells as biological cell transporters. So we're going to have the artificial cells will be these droplets. I'll introduce that topic first and then move on with the papers. And there's these are the two papers that cover this. So I'll cover, in a kind of brief way, I guess, both of these papers. They're both related to each other, as you'll see, forming a whole story. And then we could have a discussion on that. Just as this is a point of, reflection, just putting up these two references. This work involves an understanding of physics, of chemistry, of biology. And when you do this research that sort of bridges, it has to take expertise from each of these areas. In fact, it's very hard to find journals that will accept this kind of work, because specialized journals, like chemical journals, they say, oh, it seems interesting, but it's not our topic. So in fact, we had a lot of trouble finding journals who had a broad editorial base that could handle such papers. And we found a good home at Scientific Reports. It seems like they are comfortable reviewing papers that don't fit into one single box, let's say. So that's just a little reflection for you guys who are doing research that's a bit strange, let's say, like this research is. And I'm also happy that my co-author on these papers, Sylvia Holler, is part of the audience here today. So I'm sure if I can't answer your questions, I'm sure Sylvia can answer your questions quite well. So I'm glad she's here. So this is work that Sylvia and I did together the last few years. And so just quickly, I will discuss what these artificial cells are as self-moving droplets. And then we'll talk about these potential applications. For example, can you transport biological cells around using these things? And as a matter of introduction and sort of setting for these experiments, when I start talking about artificial cells or artificial biology, I like the sort of original definition of synthetic biology. Most of you know something about synthetic biology. The original definition of synthetic biology was this one, which is basically starting from the bottom up and reproducing elementary vital phenomena. So the elementary parts of living systems from the bottom up. So in this case, for our case, from physics and chemistry, that's where we're going to start. And what are these things we want to re-embody with these kinds of artificial systems? Well, it could be a whole list of things about living systems that you might find interesting. There's a partial list here. And what I am going to talk about today is body metabolism and self-movement, although there are lots of other very interesting things that could be thought of. But I'm going to limit myself to these things today. And what about this idea of artificial cells? It seems like in the last 5 years, this concept is kind of exploding in a way. There are a few labs in every part of the world now that are publishing in this area. So it's getting to be very exciting. But the concept is quite old. And the reason I bring this up is because it sets the stage for the research that I'm going to present. And as far as my research goes, I think the first time I saw someone trying to publish a artificial cell that they made in the laboratory was this guy, Bushley. Here was a zoologist who was interested in the amoeba, very much interested in the amoeba, published huge, thick books about the amoeba, and wanted to make a simplified chemical model of the amoeba and how it moves. And so what he did was he combined fresh olive oil with potash, and potash is made from when you burn wood, and the ash from the wood in the pot. So the ash that's in the pot, the potash, is what he used to run the chemistry, and that's where the name potassium comes from this, because so it's basically potassium hydroxide. And so here you're looking down on a Petri dish. This is large scale Petri dish with some fresh olive oil. Oops, fresh olive oil in it. If I can get this to behave. Go, there it goes. And so I'm going to add 2 droplets to this. The first droplet has some red food coloring in that does not contain the potash, doesn't contain chemistry. So this is a droplet of water with some food coloring in this olive oil, and there's no chemistry, so it's not active. The second droplet is blue, and it has, in this case, sodium hydroxide, and you see the difference, okay? So we're looking at liquid-liquid systems. This one it doesn't have any active chemistry, and this one has an active chemistry in it. And so you can see, even with your eye, that there's a big difference in the dynamics of these systems. And so it's basically playing with this concept that I'll be talking about today, taking liquid-liquid systems and taking something that normally doesn't do anything, if 2 separated liquids, because one's oil, one's a water base, and you activate them with chemistry. So this is a saponification reaction. It's basically making soap, but you can see it becomes quite lively. And this is what Bushley was studying when he was looking at like artificial models of the amoeba, which was quite interesting that he was doing this more than 100 years ago. And so when we look at the same system in the microscope, we see some really nice dynamics. This is the product of the chemistry, some sodium oleate, basically, something like this, and 2 active droplets of water. And we see behavior. I call it behavior, is what it seems like to me. So you have the collection of droplets, you have the fusion of droplets, you have group dynamics, you have a group of active droplets of sodium hydroxide here in olive oil again. And we see some rather surprising behavior that looks like, well, could be biology, let's say. So I think you get the point. Fascinating stuff, right? I mean, once you start animating things that normally are not animate, you start to find very, very strange things. So I'll just discuss 2 systems with you as the introduction. So we try to keep these things simple. Here's a Petri dish. We put some water phase in it, which is this blue stuff, and then we have this droplet which has a higher density than water, so it sinks down to the bottom of this thing here. So this is nitrobenzene droplet here, and this won't do anything. There's no chemistry. So we mix in with the nitrobenzene here some fuel, which is an oleic anhydride. It's you don't have to worry if you don't know about this one here. Basically, it'll hydrolyze when it gets in contact with water, but it mixes very well with the nitrobenzene. So now this nitrobenzene here is impregnated with some fuel, just like you would put gasoline in your car. And then we can see when we put it into this arena here, which is again a Petri dish filled with aqueous phase, we put this red droplet in and it just goes. It starts to move around spontaneously. So we have a self-moving system. Again, the nitrobenzene in water would normally do nothing. It would sit there, but when you animate it with this chemistry, it forms a very nice self-moving droplet. And what's happening with this color change, it's a pH indicator. That all the details for the experiment are up here. So there's a pH indicator here, which is blue at high pH and becomes colorless. So what's happening is once this thing starts to move, it starts again. We can see this chemical trail of low pH around the droplet. Right? So we know that this thing has something to do with pH. So we started playing around with that. This is the chemistry. For those of you studying biology, you'll think this is really way too simplistic to the point of stupidity that you would call this metabolism, but it is a proxy for metabolism in the sense that you have this organic molecule that becomes hydrolyzed in the system to form 2 fatty acids, so 2 oleic acids. Under high pH conditions, these are ionized, so you get 2 oleate surfactants plus some protons here. That's it. That's the spontaneous chemistry that fuels this whole thing. So what about, this is where the pH difference comes from. You're making basically acids in the system. So you would expect a pH change in the system. So here's the arena again with a self-moving droplet. And then I put in a pH gradient here with some sodium hydroxide. And then we see the behavior of the system. So this droplet, which is moving around through the system, is able to actually climb this pH gradient. So we call this a kind of artificial chemotaxis. It's not biological chemotaxis, but it basically is doing the same thing. These droplets can sense their environment and then reconfigure their motion to respond to chemical gradients. So in fact, it's chemotaxis in a very simple way with just these components here. Just not to confuse you, I should have said before, for our projects, so these droplet systems, this is an oil droplet. So that red thing's an oil droplet, and the environment is water. Okay, so it's the inverse of the Bushby system. I didn't say that, but I want to just clarify. But you can do both. You can do different kinds of systems. The ones I'm talking about are oil droplets in a big pool of water. Okay. So what's happening here? We have two things. We have sensing of the environment and we have motion. First, what about sensing of the environment? This is a depiction of one of these oil droplets, and water's out here. And the water contains the surfactants, oleate, like I talked about. That's this stuff here. This stuff. Okay, so the droplets are producing this stuff. They're surfactants. So they're going to basically form a layer at the oil and water interface, as depicted here. And these fatty acids are interesting because they only have one functional head group, which can be ionized like here, and it has a nonpolar tail, let's call it. So that's our surfactant system. And it only has two states, either it's protonated or it's deprotonated, depending on the conditions of the experiment, and most specifically the pH of the experiment. So this is what happens when you titrate the pH of these systems, looking at the tension, the surface tension between the oil and water that do not want to mix. You then have a very nice transition in interfacial tension based on the pH of the solution. When we start one of these experiments like this one here, we're at very, very high pH. So here you go, pH 12, for example. So we're way up here. And then this chemical system, as it evolves, produces acids. So the pH starts to drop. So we're basically titrating, it's titrating itself down this way. And you can see when the pH gets lower and lower and lower, that you basically go from low interfacial tension to high interfacial tension. So that means these molecules are changing their configuration based on this pH gradient that is around a droplet. For example, if the droplet is moving, oops, I lost the screen. If the droplet is moving in this direction, the acid is coming out this way, and so it's basically protonating these guys, and you're going to get very, very high tension on this side, and you'll get very low tension on this side of the droplet. That's the key to explain what's happening, because you have a reconfigurable surfactant and interface that is responding to the pH difference, and the pH difference on one side can be different than the other side because you have an asymmetric droplet. When that thing starts to move, the symmetry is broken. So therefore, you have a difference in tension from one side to the other. That's key to note, because then that gives us an explanation for what's the motor of this. So that was the sensor. What's the motor? Well, it's all fluid, basic fluid dynamics. You have a droplet here. At that interface, you have a reconfigurable surfactant, which is being protonated or deprotonated. And at high pH, you have a much different interfacial tension than at low pH. And when that happens, when you have a disequilibrium along that interface, more tension on one side than the other side, then the fluid starts to flow. It's called a fluidic instability, or more specifically, a Marangoni instability, that basically starts to flow things around in that droplet to equilibrate the tension around that thing, okay? So that results in fluid motion along this droplet, and you'll see a video of this in a moment, which basically forms a convective cell. For those of you who don't study physics or fluid dynamics, there's not too much of that here. Feel free to ask me, but I'm trying to explain in a sort of an intuitive way what I'm talking about. That the fluid starts to flow to equilibrate the tension around this, and one way you could do that is by making a convective cell. That means it starts to move. If you can see my video like this, you can see my hands moving. So you get a convective cell. And this is common. You'll find this in your kitchen when you're heating something up on the stove, like some liquid. You'll see some kind of fluid motion like this. You'll see weather patterns that move like this. So when you have physical systems that have a gradient, you often get this kind of interesting flow patterns like convection. And that's what we see on this scale as well. So that's the situation. So that's how we get fluid motion. And at these scales, these size scales, we estimate we have a low Reynolds number. What that means is that viscosity will dominate. What does that mean? That means if this droplet is flowing like this, okay, on this side is another cell on this side, so you have this convective cell going. The surface of the droplet moves and is in contact with the water and forms friction. So it's basically forming friction in a viscous environment. That physical friction that it forms when it moves its surface is enough to propel itself forward. This is the same thing that happens when you swim. When you swim, you put your hands out flat and you push with your hands because you want to make as much friction as possible with the water around you. So that's what this is doing. It's making friction with the water. So it's a swimmer, basically, is how this thing is working when it moves from place to place. So I hope that explanation was clear. So that's it for the fluid dynamics. So here is biological chemotaxis that you guys probably know quite well. In biology, you have cells that have specific receptors for small molecules, in this case, cyclic AMP. They then reconfigure their cytoskeletons, their actin, for example, to move directionally towards where the source of chemistry is. For example, coming from this pipette here, you can control the motion of a dictyostelium cell quite well. Because of basically evolution, biological evolution has produced this wonderful kind of behavior. What we're showing with really non-sophisticated droplet systems, like on the left here, is we can do something similar with controlling the behavior of these droplets because they can sense their environment and they can move in that environment. So we could also have chemotaxis. And again, this is chemical chemotaxis, has nothing to do with cytoskeletons. There's no DNA, there's no protein, but yet we can do it. We can reproduce this kind of phenomenon in these artificial systems. So that gives you an idea, I think, of the flavor of these kinds of artificial cell research projects. Oh, here we got, this is the video of, here's the droplet again, that's moving, saw it before. Now here are these droplets that are convecting. I hope you can see that each of these is convecting slowly. You have a center line and then you have this convection around each of these as this moves. I just wanted to show you this. So you have a sensor, you have a motor, they're coupled together. And when this droplet moves, basically it's moving away from its own waste products, right? Which is this, basically the fatty acids that it's producing. And as long as it can move away from its waste products, it's going to prolong its approach to equilibrium. It'll still find its way to equilibrium. It doesn't have any more sophisticated mechanism. It's not able to eat like we are. We're able to eat to avoid equilibrium. This droplet is not eating anything, but it is moving itself away from its waste products. So in a chemical sense, it avoids equilibrium, prolongs its approach to equilibrium. And perhaps it's also performing some sort of self-preservation, which is more of a philosophical argument about this. But this is what we're thinking about with these chemical systems. Okay, here's another system, and this is going to form the basis for the rest of the talk. So it's just it's simpler, really simple system. You have another big dish here. You put some water in it, and you put another droplet in. So that's all the same. This case, we changed what the droplet is. It's 1 decanol. It has a density lighter than water, so it's going to float at the top. And we don't put any fuel in it this time. So that chemical reaction saw before is now gone. And this droplet won't do anything. It's really even simpler than that previous system. It won't do anything unless you perturb it. So it's kind of like, I don't know, it's kind of like something floating on the water, and then you can get it to move by doing something to the environment, like pushing it in a certain way or whatever, like this. Or you have something on wheels that's not moving. You can push the thing on wheels and it will move for some time. Or you can change the gradient of the landscape by tilting the landscape, and your ball will roll downhill. So it has the potential to move, but it doesn't have its own energy anymore. This is a simpler system, has no fuel in it. But we can do cool things with it. Like this is a microscope slide here. And what's going to happen is we're going to put one of these decanol droplets on this side, and then we're going to put in a chemical gradient on the other side, and we're going to look at the time evolution of this particular experiment as far as what happens. These are the chemical details here, but here's our droplet of decanol. Salt is going to be added here at some point. Right there is just added. Now it's going to take some time for that salt to diffuse across this slide here to reach that droplet sitting there. There it goes. So even these simple systems, this is simpler than the one that was quite simple before. Basically, yeah, these things are responsive again to their environment, and they can move through fluid dynamics from point A to point B, so it could follow chemical gradients. So that's great. And the reason is because of surface tension again, because we're then monitoring, what's happening with this decanol in different kinds of circumstances. And when you add this, I didn't say what this gradient was. The gradient is sodium chloride. So if I go here, just using salt, sodium chloride. That's all we're using here. So the salt, this droplet is attracted to the sodium chloride we added over there. And that's because when you titrate in sodium chloride, you again change the surface tension landscape. So it's very similar to what I just talked about with the previous one. These droplets are basically looking to equilibrate themselves, and they equilibrate themselves where at the lowest surface tension in the system. So that's at the highest salt concentration. So it will move itself to the highest salt concentration because that's the lowest energy state of the system. I hope that makes sense to you guys. And so you could do funny things. This is a nice funny thing we did by putting decanol droplet here and putting the source of salt over on the other corner and seeing if it can solve a two-dimensional maze. So that's the salt. The salt is impregnated into this droplet here. That one doesn't move, but the pink one is moving. So you get the idea, right? So yeah, it's a lot of fun to do these experiments, as you can imagine. By the way, it's also quite cheap to do these experiments. I mean, the chemicals are real cheap. And by the way, this is just a glass slider with some packing tape, to make these little barriers. This is a good experiment. It doesn't take a lot of time or money to invest in something like this. But that's the proof of principle. Again, it's play it one more time. So this droplet, it's not doing anything until there's a salt gradient added to the system, which is coming from this one. The salt is slowly diffusing through these lanes here, where this droplet can then sense them. And if it can sense the salt gradient, it'll move again to the highest concentration of salt, because that's where the lowest surface tension is in these experiments. So from our point of view, we see a droplet solving a maze, but probably from the droplet's point of view, it's just basically looking for energy minimization. It's just finding the lowest energy state of the system. And for that droplet means move this way, move this way, move this way, and come here. I mean, that's it. From our perspective, it looks more complicated, but probably from the droplet's perspective, it's really, really simple to solve this maze. Okay. So that gives you intro to these self-moving droplets. And now we're going to talk about, well, see, the problem is when you try to, again, publish these things or write grant proposals, people are always bothering you. What are you going to do with these things? Well, the honest answer is we work with these because they're a lot of fun. And they're interesting, and they surprise us, and they challenge us to think in ways that we haven't really thought before, which is, I think, that's the real reason. But there have been some efforts then to show you can do something quite cool with these kinds of systems, perhaps as potential applications. So that's what Sylvia and I did. One thing we initially did was say, well, can it move stuff around if you put stuff in it? So here's the same system, it's a chemotaxis system. Here's our droplet. And there's a little piece of copper wire that we placed inside of there. And then we put a chemical gradient, I think over here. There it is. Here's a salt gradient. So we were able to move the droplet from point A to point B. And now what Sylvia is doing is adding a decoupler. It's just a bunch of soap. And he's trying to tell the droplet to let go of that thing. And it didn't let go yet, but it moved over here. And now she's telling it, please let go of that copper wire and then move over here. And finally it listened to her, and it was able to basically move that little piece of copper wire around. So that was just a little proof of principle experiment too. That we can move stuff. But can we move living stuff? This is the challenge. Copper wire was easy. we need to make this artificial chemical system compatible with cells, compatible with the decanol system, so the chemistry of the system. We want it to be coupled and then decoupled from the droplet. So not just move something from A to B, but then leave it at point B, leave it there and move out of there. And we need to somehow get the cells out of the system, and hopefully they're still alive. And if you followed the introduction so far in the previous part, you understand that, well, cells really typically don't grow at pH 11 or 12, for example, unless you had some extremophiles, which we don't have. In fact, don't have extremophiles, just have normal laboratory cells. So this is an issue, because when you put cells in here, they immediately will die. I mean, really immediately. You know, just 1 minute later, Sylvia assessed if we can get any, if anything is still alive, and no, everything is dead. So this is a good way to kill cells, by putting the living system in with the artificial system. Everything dies. So really, we had a problem. So one thing that you guys know from tissue engineering is you can use different kinds of matrices to interface in a very kind of commensurate way with living systems. For example, using hydrogels. So we use alginate hydrogel. This is very standard. And, you know, there are lots of people using this for all kinds of cool things, these things. So we wanted to use hydrogel as a way to package our living cells, put it in the artificial system to protect the cells from dying during this whole experiment. So that's the situation. So here we go. Here's the same decanol experiment. So Sylvia is going to place with tweezers a little package of alginate, we call it capsules, alginate capsule in there. And then she's going to put in a chemical gradient with sodium chloride over here, and she's going to see what happens. There's our little, see a little white thing. It's our package. She made the chemical gradient. There's the chemical gradient, and there you go. Droplet moves perfectly, leaves the package there. And those of you who know about tissue engineering can immediately understand why. Is because, you know, this is a hydrogel and does not want to interact with non-polar substances, right? It wants to interact with water. So there's a physical problem using hydrogels that we want to interface stably with a non-aqueous phase, non-polar phase. So we then had to reconfigure the system to be compatible with our droplet, with the decanol, which is mostly non-polar. So there are ways in the literature where people have reconfigured alginate or other hydrogels chemically to make them more non-polar. And I don't know, in my lab, I always like to think of really cheap and easy solutions to this. Instead of resynthesizing alginate, which sounds like a pain, I want to do something similar, but cheaper and easier. And so we came up with this solution, which works pretty well. So what you're looking at here are some contact angle measurements for hydrogel. That's the horizontal slab here in all of these. It's the hydrogel, same as this. And something has changed between A, B, C, and D. You can see the contact angle is changing, right? That means this hydrogel is becoming more and more hydrophobic over time. And so we're not changing the chemistry of the alginate. What we are doing is just adding in a surfactant to the system. So this is just some measurements with water. And here's our surfactant called decanoate. It's basically monocarboxylic acid of 10 carbons, all at the same pH. And you just titrate in more and more amounts of this very simple surfactant. And it basically then changes the chemical property of the alginate, which is great. So easy. All you do is when you make your alginate, throw in a little bit of surfactant, and you can modify the contact angle and the sort of hydrophobicity of your hydrogel. So that's works really well for us. So that's what we did. We basically made our hydrogel in the presence of some surfactant to make it more hydrophobic, so that when we have our two decanol droplets here with two packages of hydrogel, and we basically want them to come over here, you can see then what happens. This time droplets move towards move towards the source of salt, and they take their packages with them. So now they have an association. Now, can we break the association? This is what Sylvia is trying to do by adding in a decoupler. So lots of surfactant to say, please let go of those packages and move out of the way. So there you go. So the delivery was made, and now the delivery people get out of the way. That's really what we want. So we're able now to transport and release these hydrogel capsules. But what happens with cells? Can we actually make cells sort of comply with the rules of this game or not? And so we tested a bunch of things. Empty capsules, that's like what we just saw, or capsules containing different kinds of living cells. So Vibrio fischeri, E. coli, Bacillus subtilis, and yeast. And we then tested, first of all, can you transport these different types of organisms? And then when you do the transport, want to check for a few things. Is the transport sterile? Is it aseptic? And are the cells alive, and do they actually proliferate after this whole ordeal, this whole test? So this is how we set up the experiment. So it's similar to the video you just saw. 2 droplets, 2 different capsules inside. And one, for example, could be an empty capsule containing just growth media. And the other one would be made with growth media and living cells. And then you add the gradient in point B, and you hope that the transport will happen. Bring the packages with them, add the decoupler, so it says, please leave the packages there, and then set up a second gradient, C, to move the droplets out of the way. That's basically the video you just saw. And then we go in, take these capsules out of the experiment, and then assay them if the cells are still alive and still basically growing. And so this is just some plates of these. Here's capsules of E. coli, a subtilis here. These reproducibly survived the experiment and proliferated after the experiment. So these kinds of lab strains seem to be okay with being encapsulated in the hydrogel, the modified hydrogel, and transported in this experiment. But not everything was happy. For example, Vibrio fischeri here, by the way, when you directly add them to the chemical system without the packaging, they die. They survive the encapsulation in the hydrogel, but they do not survive the transport, which could be just a few minutes in the system, and Vibrio fischeri is dead. E. coli and Bacillus subtilis did better. They grew no matter what was happening, they were fine. It appeared that all of our organisms here did not like that decanol drop. But again, if you put in decanol without the capsule, without the protection, they die, they're gone. The surprising thing was that yeast did survive these steps, but it was not consistent enough. So we weren't able to get very good quantitative results from that, like we could get with E. coli and B. subtilis. But yeast sometimes survived this whole system, and sometimes was not really happy with us at the end of this. So those are it's just a summary of that. This is another summary of these transport properties. So yes, the transport was sterile because that environment is so harsh that no matter where you sample outside of that capsule, you find no viable cells. It is aseptic, which means when you're transporting, well, if you're transporting 2 droplets, so this one does not contain cells, but contains growth media, and this one contains growth media and cells, there's no transfer of live cells through the system from one to the other. You cannot cross-contaminate the system. They are really isolated. So the aseptic conditions were good. And yes, we did see some cell viability and proliferation after transport, but not every organism will tolerate this. Just some organisms tolerate it. That's the lesson from that first paper. So moving on to the second paper. We're going to continue the same system, but we're going to make it harder. So what about human cells? Can we do that? So here's the overall scheme, which you're probably familiar with by now. So you just grow up some human cell lines here. You encapsulate them in one of these hydrogel things. You incubate them in DMEM or some growth medium like this. You put in your droplet, and then you place one of these capsules on it. These are the kind of orientation of the surfactants in the system that we add to put all this together. Remember, if you didn't put surfactants in the system, the capsule doesn't stay associated with the droplet. So you need to put surfactants in the system to make this work. And you need to put surfactants in the system for that surface tension difference to make the thing move, because that's what's responsive to that chemical gradient. So you need the surfactants both for the association of the capsule with the droplet and for this chemotactic motion if it's going to occur in the system. So you need that for both. So we add in the surfactants, you put your droplet with your package, you put in the chemical gradient, and then you test to see, does it move? Does it transport the package? And then you move this out of the way and you harvest your packages, you harvest the cells, and you see, are they viable and are they metabolically active? So are they still okay human cells after this same ordeal as we put the other ones through? So here's just some results from that. So we have some bar graphs before transport, after transport. So let's start with this one, before transport. So this is cell viability. So this is, for example, tryptophan blue test here, MTT test for metabolic activity here for 3 different human lung cell lines. The viability means you package them in the hydrogel and then you take them out and you see if they survived. So yes, they do. You didn't transport these; you just packaged them and unpackaged them, and you look to see if they're still alive. And you can see, yes, by and large, we still pretty good viability. Not 100%, but we still have some activity from these cells. So they're not completely happy, but they're not so bad. This is just a control, the same 3 cell lines, same 3 assays, but we bleach treated them. So we really gave them the chemical treatment. We just throw bleach on them to kill them. And you can see that that harsh chemical treatment, which we don't have in the normal transport experiment, that is enough to really knock them down. Then package them, transport them, then harvest them, and you can see what happens here. Not all cell lines behave the same. For example, the A549 cells are not happy and probably not going to proliferate very well, if at all, after packaging and transport in the system. But these other human lung cell lines appear to be quite good. Basically, they survive the transport. They're about the same as when they were packaged. So yeah, so the system works. It is able to transport human cell lines, and some, but not all, of the human cell lines will still survive this kind of test. But then we saw something quite interesting happening, and it has to do with the name of this paper that we published here. So I talked about transport of mammalian cells. We talked about the chemotactic droplet, but we added this word in, which is key, autoselective. What does the autoselective transport of cells mean? Well, what that means is this scheme that I showed before now has been modified. Everything is the same, except we're not putting in surfactant into the system. Now I told you, first of all, you need surfactant to make a stable association between the droplet and the package hydrogel. That's number 1. You need surfactant to have chemotaxis happen from point A to point B. So we actually know we need surfactant because we've done all the tests. We need it in order for that system to work, but we take it out. So we just take the surfactant out of the system. So now the system somehow needs to, if it can, it needs to supply its own surfactant. You could probably imagine what would happen if you're using human lung cells, because human lung cells can produce biosurfactants. That's what we want to understand. And human cell lines can produce biosurfactants if they're alive. If they're dead, they're not producing them for us. So what we're looking for is a system that is autoselective for living cells that are producing biosurfactants. So can this system be selective? And that's what we then tested. So here are so all of these surfactants that we see in the system that are necessary are not being supplied by us anymore, but they have to be supplied by these cells if they are alive and if they can produce the biosurfactants. So here are the results of this. So what are you looking at? So this is the velocity of the transport and the efficiency of going from, let's say, point A to point B. So that means this group of conditions were not basically not working. These don't work, they don't move, and there's no velocity basically, and there's no efficiency of going from A to B. So these are the non-working group. This is the working group. All of these conditions worked. These were able to transport the cells. What are the conditions that worked? Well, these colors correspond to these. This is where we added in the decouplers. We added in this surfactant. So we know that the system needs this to be active. So those are all represented here. But there's one point on both of these graphs where we did not add the surfactant. These lung cancer cells are there, and they are moving and being transported without us adding in the surfactant. They are supplying their own biosurfactant in order to associate with the droplet and to be transported by the droplet. Interestingly, these cells are also here alone, and they are not encapsulated in the hydrogel, and then they don't work. So the same cells that allow for this successful transport need to be encapsulated in the hydrogel first. So that's what this says. And if they're encapsulated in the hydrogel, then they produce biosurfactants, and then you could move them in the system. If you don't package them in the hydrogel, they don't move. This means, by the way, the capsule here means they were not encapsulated. It's just that we put in alginate capsules, just put them in. The cells were not part of the capsules. We just put them in the experiment as a control. The cells were just free living in the DMEM media here. So these don't work. You have to encapsulate your lung cells. They start produce biosurfactants. They then associate stably with the droplet, and then they're moved, and they survive, and they survive the transport. So this is the system then as a whole becomes selective for living cells producing biosurfactants. That's the conclusion from this. Okay, so just some conclusions and then a few notes, some advertisements at the end. This is what we are interested in, coupling chemistry with these kinds of fluid systems to produce cell-moving systems. We love chemotaxis, for example. We like to do fun things like solve mazes, use these as transporters, as my colleague Yitka Chegova says, a chemotaxi. You can take this around. You could leave your passenger somewhere and get out of the way. The cool things I think about this are looking at how simple chemical systems can be responsive to their environments, including environments that they're self-modifying, by the way. I think that has a lot of potential. So you can see systems that act and react with their own environments. So you have some crosstalk, there's information transfer between these artificial systems and their environments. Perhaps they have this emergent mechanism of motion to avoid equilibrium, or prolong equilibrium, I should say. And we're investigating how we could bring these two very different systems together, living systems and artificial systems, to complement each other to produce a coherent system, as I was talking about in the last part of the talk. For those of you who are interested in these kinds of things, I just have three little advertisements. One is we have this, as Antonio mentioned, a European project with artificial cells where we're making these kinds of droplets with different kinds of biochemical functionalities to make artificial cells. It's called ACDC here. We have a second project, which is an educational Erasmus project. We bring together art, design, robotics with artificial biology. This is our contribution here. How all of us can work together to produce these kinds of systems. Maybe you might consider them to be systems of agency or even cognition. Perhaps we can explore this area. So this is an active area of investigation. And if you really like this weird kind of science, I'll encourage you to check out the conference on artificial life, which will be summer next year. We are hosting it, but it'll be virtual because we are worried that no one will be able to travel and we'll have to cancel everything. But it'll be online, so it makes it easy for all of you to join if you're interested in this conference on artificial life. We don't even have a website yet, unless Sylvia corrects me, but you can look for this for next year. Okay, thank you for your time. That's it. 45 minutes.
Very nice presentation.